So we'll start the endoplasmic reticulum. So here when you take endoplasmic reticulum and designate ER, okay, endoplasmic reticulum. You take endoplasmic reticulum. See, if you assume that this is an animal cell, which at the center you will be having nucleus, cytoplasm, and different cell organs will be there. Now, from the nuclear membrane, around the nucleus you will be having nuclear membrane, like the plasma membrane around the nucleus. This nuclear membrane, and this is the plasma membrane. Okay. Now, when you take animal cell, I'm insisting animal cell. When you take animal cell, there is a network of structures between nuclear membrane and plasma membrane. Okay, we're using the word network. Network of structures. What are those network of structures? We'll see in detail now. Network of structures between nuclear membrane and plasma membrane. So what are those network of structures? Two cell organelles. One is endoplasmic reticulum, another one is Golgi body or Golgi complex. Okay, so see the diagram here. So the purple one is the nucleus, nuclear membrane. Okay, and this is the plasma membrane. Part of a cell they have shown. Now from here to here, just showing with a bracket, here to here, it is endoplasmic reticulum. Don't bother about rough. Endoplasmic reticulum, this part, the yellowish part. And from here to here, showing with another bracket, that is Golgi complex. So here, when we talk about the nuclear membrane and plasma membrane, in between them, there will be spanning of network structures of endoplasmic reticulum and Golgi complex. Remember this point. This carries much importance in further discussion of the class. Okay. Now we are going to discuss only about endoplasmic reticulum. Once we get finished with that, we can go for Golgi complex. So both are network structures between nuclear membrane and plasma membrane. The rough diagram I have given. Nuclear membrane, plasma membrane. In between you are having a network. This network includes endoplasmic reticulum and Golgi complex. Now we'll discuss about endoplasmic reticulum in detail structure first. When we take endoplasmic reticulum as I told is a network of structure. You'll find endoplasmic reticulum only in eukaryotes. So endoplasmic reticulum is absent in prokaryotes. Another statement. When you take endoplasmic reticulum number, in a cell, endoplasmic reticulum may be in any number. See, this is an animal cell where endoplasmic reticulum was shown here and even shown above also. See, the same yellow one, shading on that. So in this cell, you are having two places endoplasmic reticulum. And in some cells, more endoplasmic reticulum you may find, more. Okay. Bhumika, do you have any doubt? Okay, network issue, no problem. So more endoplasmic reticulum where you will find? You will find in actively dividing cells. The actively dividing cells or actively growing cells will have more endoplasmic reticulum. So that's about endoplasmic reticulum importance. Now when you take endoplasmic reticulum, as I said, it is a network of structure. It possesses three parts. Cisternae, tubules, vesicles. These are the three parts which make up the whole endoplasmic reticulum structure. This is the whole endoplasmic reticulum structure shown in elaborate way. Now when you take cisternae are sac-like structures. Sac-like structures here you can see, these are the cisternae. Okay, this is another cisternae. This one would be another cisternae. And tubules are the tube-like structures which joins the cisternae. So this cisternae and this cisternae were joined by tubules. shading the tubules. So that would be the tubule. This would be the tubule. Tubule of one cistern and another cistern. So like that, tubules are the tubular structures which join cistern. When it comes to vesicles, vesicles are the part of endoplasmic reticulum which are separated from the cistern and tubule network. As we are saying, the cistern and tubule will be in network. See, this is all in a network-like condition. Interconnected. Network is nothing but joined. But vesicles are not joined to cistern and tubules. They are separated out. So these are the vesicles, the separated out bubble-like structures of endoplasmic reticulum. So when you take the endoplasmic reticulum, you can justify its structure by concluding that endoplasmic reticulum is a network of cisternae, tubules and vesicle-like parts. These are different parts, nothing but different parts of endoplasmic reticulum. So roughly if we want to draw the endoplasmic reticulum, you can draw like this. So the rough way I am drawing it. So roughly if you see a diagram like this, it would be endoplasmic reticulum with cistern and tubules. Okay, the same thing I have shown here in elaborate way. Now what are the parts of endoplasmic reticulum? Cisternae, tubules and vesicles. These are the three parts of endoplasmic reticulum. Okay, now when you take the endoplasmic reticulum based on presence or absence of ribosomes can be divided into two parts. Ribosomes, what are they? I'll discuss. Endoplasmic reticulum can be divided into two types based on the presence and absence or absence of ribosomes. If ribosomes are present, then it is called rough endoplasmic reticulum. If ribosomes are absent, then that is called smooth endoplasmic reticulum. Okay, so here they have shown the ribosomes dotted-like structure here. See this diagram. So on the cisternae, on the vesicles, and even on the tubules also, you have dotted-like structures. These are ribosomes. That's why this part we call as rough endoplasmic reticulum type. Apart from rough endoplasmic reticulum, you will have smooth endoplasmic reticulum also, where you won't find ribosomes. Ribosomes are absent. So based on presence or absence of ribosomes, endoplasmic reticulum may be rough and smooth. Two categories of endoplasmic reticulum. Now what are ribosomes? Ribosomes are nothing but the cell organelles. They are present in the cell only, that's the cell organelle, which are involved in protein synthesis. Okay, so based on the function of ribosome, if you take the function of ribosome, what is the function of ribosome? It produces proteins. So indirectly you can say rough endoplasmic reticulum produces proteins. Smooth endoplasmic reticulum cannot produce proteins. Got the point, Anna?
Yes, ma'am.
Yeah, Bhumika, definitely I'll explain. Anna, you got the point about what the statement I gave. Rough endoplasmic reticulum is involved in protein synthesis. Smooth endoplasmic reticulum is not involved in protein synthesis. How you will justify? Because of the presence and absence of ribosomes. Okay. No problem, Bhumika. I'll explain. See, that's why I didn't erase anything. Yeah, I know. Vinay sir initially told me from the past few days you are having connectivity problem. That's not a matter. A simple point only here. The endoplasmic reticulum, if you take endoplasmic reticulum is a network of cell organelle. You will find endoplasmic reticulum only in eukaryotes. You won't find it in prokaryotes. In prokaryotes, it is absent. Okay. Now, when you take the endoplasmic reticulum, in this animal cell diagram, there are few endoplasmic reticulum. Here one and here one they have shown. But you will find more endoplasmic reticulum in cells which actually divides and grows. Okay. So, when a cell is in active growing stage, the endoplasmic reticulum will be more in number. That's the point here. That's about endoplasmic reticulum. When we take endoplasmic reticulum structure, the structure of endoplasmic reticulum includes 3 parts. Okay. The first part, cisternae. Second part, tubules. Third part, vesicles. Okay. Now, when you take cisternae are sac-like structures. So, here in this diagram, the endoplasmic reticulum is shown in elaborate way. I'll use another color so that it will be clear for you. Here. So, I'm showing with a green sketch. So, this is sac-like structure of endoplasmic reticulum. This is another sac-like structure of endoplasmic reticulum. Likewise, you are having sac-like structures of endoplasmic reticulum. So, these are called cisternae. Cisternae. Remember this word because you will get cristae in another cell organelle. Okay, cisternae is related to endoplasmic reticulum. Then tubules are the tube-like structures which will connect different cisternae. Here, if this is one cisternum and this is another cisternum, both are connected by tube-like structures. Shading with blue so that you will differentiate. So, these tube-like structures which are connecting the cisternae are called tubules. Okay. Then vesicles. Vesicles are not in the network of endoplasmic reticulum. Vesicles are the part of endoplasmic reticulum which are away from the cisternae tubule. This is vesicle. Likewise, this can also be concluded as vesicle. It is away from the network of cisternae and tubule. So, this is how endoplasmic reticulum can be shown as a network of 3 parts called cisternae, tubules and vesicles. Okay. Now, this rough diagram of endoplasmic reticulum if you draw. So, these are the tubules which are connecting cisternae. Individual cisternae we are connecting with tubules. And vesicles will be separate like this. Not in endoplasmic reticulum, cisternae and tubule network. This could be vesicles. So, these are the 3 parts of endoplasmic reticulum. Now, when you take endoplasmic reticulum can be categorized into 2 types. Types I am talking. Based on the presence or absence of ribosomes. If ribosomes are present, then it is called rough endoplasmic reticulum. This is rough endoplasmic reticulum. See, all over you are having dotted structures on cisternae, on tubules, on vesicles, everywhere on endoplasmic reticulum you are having dotted structures. These are the ribosomes. So the presence of ribosomes will give a one type of endoplasmic reticulum called rough endoplasmic reticulum. If ribosomes are absent, then they are called smooth endoplasmic reticulum. When you take ribosome function, ribosomes are also cell organelles that are involved in protein synthesis. So, rough endoplasmic reticulum is involved in protein synthesis, whereas smooth endoplasmic reticulum is not involved in protein synthesis. Why? Because in rough endoplasmic reticulum only ribosomes are present, so they are involved in protein synthesis. This is all about endoplasmic reticulum structural aspect. Clear, Bhumika? You have any doubt? Okay, fine. Then, after smooth and rough endoplasmic reticulum differentiation, we'll move on to next slide. So, that's what I just explained, rough endoplasmic reticulum and smooth endoplasmic reticulum. And these points we'll discuss in structural functional aspect. Sorry. Now here, this you can see cisternae. So cisternae are these things, the folded ones, and they're joined by tubules. Okay, circling. So this is cisternae, and those are the tubules, and the dotted ones are the ribosomes. So this is rough endoplasmic reticulum, and This part, I'm making a circle in the diagram, see. So this part, it is not having any ribosomes on it. So it is smooth endoplasmic reticulum. The labeling was somewhat blurred to me. I don't know how you got the slide. This is smooth endoplasmic reticulum without ribosomes, and this is rough endoplasmic reticulum with ribosomes. Okay, clear? Shall we move on to next slide? The remaining points in this slide are the functional aspects we'll discuss now. Now, this is about the rough endoplasmic reticulum. Just a moment. Okay. So here, when we take the rough endoplasmic reticulum, they have given as RER, stands for rough endoplasmic reticulum. Now here you will find ribosomes. So we are saying that the ribosomes will be there on the rough endoplasmic reticulum. How the ribosomes get attached to rough endoplasmic reticulum? By means of proteins called ribophorins. Ribophorins are the proteins which helps in attachment of ribosomes to rough endoplasmic reticulum. Okay. Now the ribosomes will synthesize proteins. Now here the point comes, we are discussing proteins all the way rough endoplasmic reticulum. What are these proteins actually? Here there's a one point here. See, I'm circling it. Precursor. Do you know what is meant by precursor? Anyone of you? Precursor.
No, ma'am.
No. Fine. Precursor is nothing but the initial molecule. I'll take an example. Suppose you're having an enzyme. Enzymes are proteins. This you know. I think all enzymes are proteins. Are any enzyme is non-protein in nature? All enzymes are proteins, or any enzyme is non-protein in nature? You would have it in chemistry, isn't it? You'd have read the apoenzyme, coenzyme, prosthetic group, cofactor. You'd have read
No,
it.
ma'am.
No. Both of you 9th standard only, no? Then CBSE only.
Ma'am, we are in CBSE.
CBSE only, yeah. Then, yeah, that's okay, no problem. So when you take enzyme, now I will explain in very simple way what is meant by precursor, so that the protein synthesis topic you will get an idea. Now take enzymes. Suppose this enzyme is acting on a substrate and converting it to product. This you know. Now the enzyme has to be active, isn't it? The enzyme has to be active, and this enzyme will make the substrate to get converted into product. If the enzyme is inactive, it cannot act on substrate, so that you will not get product. Is that point clear? Active, inactive nature of enzyme. So far that point is clear. Is that clear? Active enzyme converts substrate to product. Inactive enzyme cannot convert substrate to product. Now here precursor is nothing but the inactive enzyme. For example, I am telling here. Inactive enzyme is the initial molecule. Until unless this inactive enzyme come across with this reaction, it will be in inactive form. Once it gets involved in this reaction, it will make the substrate to get converted to product. So the inactive enzyme is converted to active form. So what is the previous form? What is the initial form? That is called precursor. So what rough endoplasmic reticulum will do is, it will produce enzyme precursors. Enzyme precursors are nothing but the inactive form of enzymes. When the inactive form enzymes will get active, when they are involved in conversion of substrate to product reaction. So such kind of enzymes all are protein in nature. All the enzymes which are involved in digestion, the enzymes which are involved in circulatory system, the enzymes which are involved in excretory system, all those enzymes are protein in nature. So all those enzymes are synthesized by ribosomes, and some ribosomes are present in endoplasmic reticulum, making it rough endoplasmic reticulum. There's a connectivity. Try to understand the connectivity. You got the connectivity? Shall I repeat the connectivity point again? Yes, are you done? Shall I move on, or shall I repeat the precursor point and protein point again? Yeah, fine, Bhumika. That's what. See, wherever you find ribosomes, this ribosome will follow you throughout the topic of cell. Okay, wherever you find ribosome, ribosomes are involved in protein synthesis. Why ribosomes protein synthesis is so important? Because proteins are the one which are acting as enzymes in your body. In animal cell, for example, human body I am taking. Okay, all the enzymes are mostly proteins. Some enzymes are there which are not proteins. We are not bothered about that here. Now, these enzymes, they are involved in digestion process. Means the proteins are involved in digestion process. They are involved in excretory system. Means the proteins are involved in excretory system. Likewise, various life processes are linked up with proteins. So, in order to carry out digestion or excretion or any other process, you need proteins. You need enzymes, and definitely enzymes are proteins. Okay. Now, when we take proteins, the proteins will be produced by ribosomes, and we are discussing here about synthesis of precursors and the ribosomes present on rough endoplasmic reticulum. Now, the rough endoplasmic reticulum contains ribosomes. So, rough endoplasmic reticulum produces proteins. How the proteins are produced? They are produced in the form of enzyme precursor. Precursor. Precursor term stands for initial or the starting point, you can say. Okay. So, the enzymes produced by rough endoplasmic reticulum will be initially in inactive form. We're talking about enzyme. Okay, enzyme is nothing but protein. Indirectly we are saying protein, but they are nothing but enzyme. Enzyme produced will be in inactive form called as precursor proteins. Okay, so initially we are saying inactive. So finally what they will do? Finally they will become active. So when they will become active? When a substrate is converted to product. So all the time your digestive enzymes will not be released. Suppose if you take digestion process, only when we have intake of food, then only digestive enzymes will be released in more number. So until then, what is the role of digestive enzymes? Where they will be? They will be in precursor form. Now you got the connectivity? They will be in precursor form. There will be in inactive form the digestive enzymes. All the proteins will be in inactive form. Once you have taken in lot food, it has to be digested, and then the proteins which are in inactive form will get changed into active form. Okay, That is meant by precursor. Precursor is nothing but initial, a starting form of inactive enzymes. Is that clear, Bhumika? Arnav, do you have any doubt?
Ma'am, I didn't understand formation of glycoprotein.
Glycoprotein I didn't discuss. I discussed only uh, the surface of ribosome point, surface for protein synthesis, synthesis of precursor, because these two are interrelated. That's why I took this point. Glycoprotein is different. We'll discuss that in the next session. Because glycoprotein discussion is something related to smooth endoplasmic reticulum. We'll go in continuation with that. Now, we discussed about rough endoplasmic reticulum functions now. Next comes smooth endoplasmic reticulum. So, in that purpose, glycoprotein synthesis is related. Okay? We'll discuss these 2 points in the coming session. Is that clear? So, almost 8:30. Shall we end up the session? Got it, Bhumika? Okay, fine.